to shoot your hand off if you say, hey, wait, I don't remember that, or I don't get that, or can you go over that in more detail? The reason why I start with hair is, as I mentioned, spring semester is a lot of biological materials. So hair is one of the biological materials we'll be working with. Right after hair, we're going to jump into the blood lectures, because same idea, a variety of different physiological fluids, but we're going to deal with the most commonly encountered biological fluid in forensic science, blood, and talking about all different aspects of it. Then we're going to move on to how do we identify blood after we have already talked about all the different characteristics. I want to do a little bit of ABO stuff with you guys as well. And in fact, as a preview, I'll remind you again and again in the next couple of weeks. If you could send me your ABO blood type, I'm going to do an extra credit exercise with you guys where when I talk about how Landsteiner discovered the ABO blood groups, we'll talk about how he did it. But you guys are going to reenact, literally reenact. But basically, I'm going to give you a spreadsheet with biological cell reactions, and you're going to make an interpretation of what blood types are of your other classmates based on those patterns. And of course, with privacy, you can give me a fake blood type if you want. Just make sure you know. Remember what you sent to me. It's got to be A, B, O, or AB. Don't give me any other weird letters or weird combinations. Brianna's will be, BP. Or True. I don't need your RH type because that won't be part of that particular exercise. But like I said, if you know your blood type in the ABO system, email it to me. If you do not know your blood type in the ABO system, you don't need to run out and find out what it is. Make one up and send it to me, and just keep that in mind because I'm going to be using the blood type you send to me to put together the spreadsheet. And like I said, when the time comes, I'll make that available, and then you guys will be able to have an assignment with the Landsteiner thing. Years ago, it was actually something that we did in lab, where we pricked fingers and you separated out your cells from your serum, and then this is exactly what Landsteiner did with his, his lab. The people who worked in his lab, that's how he discovered the ABO system. He would basically ask people, okay, let me take some of your cells, clean cells, which means that the serum was washed away from the red blood cells, and mix it with someone else's serum, and sometimes you'd get this clumping occurring, and sometimes you wouldn't get clumping occurring. So we actually used to do that up until we came to the new building here, and everything changed in terms of people being really nervous about, I guess, people pricking their fingers, and, after blood, you guys will do a physiological fluids exercise, so it's not just blood. I'm going to be talking about all manner of physiological fluids, but concentrating on the ones that are probably more commonly found in criminalistic situations. And yes, way back, before we came to the new building here, people used to BYO physiological fluids. And you always had to ask somebody else for the other thing that you didn't have, because that was the situation. Yes, we had to do that. And, because that's real. You end up working in, let's say, the OCME biology department, and maybe it's a sexual assault case or something like that, and there's someone's underwear, or is there seminal fluid on the sheets over here? That's the kind of stuff that you deal with. Criminalistics is not everything sterilized and clean before it comes to your lab bench. So you had to deal with all those things. And when I talk about, like I said, when I get to that lecture, I'll talk about the pros and the cons of not bringing your own. We purchase physiological fluids now. You have a kind of everyone gets the same stuff from the tube that we purchased, which is not quite as good as realizing the range of different kinds of responses you get in your chemical reactions, because your urine sample might be a little different from your classmate's urine sample. So you might get like a different shade of a color compared to somebody else. And that would be perfectly normal, which you don't necessarily see that when you all have the same exact sample from the same exact source, and that kind of thing. And I remember, I think the first year that we did that here, they purged the seminal fluid, but it had no sperm cells, which is kind of annoying because it's probably one of the first things you want to look for when you have, especially a standard seminal fluid sample. Not to say that there aren't situations where it will be seminal fluid, but there's no cells. But when you're learning, it's a better idea to be aware of what a more typical sample looks like. So all those issues. So by the time we get to like last year, you will have a photography exercise. So like I said, and we've reintroduced this semester the wood lab. My colleague, Dr. Peter Diachuk, has rejoined the faculty at John Jay College. He's a firearms guy, but he's not just a firearms guy, he's also an expert with wood analysis. So he will be doing the wood lecture later on in the semester. And he's not teaching in the lab this semester just because, as you may have seen already, we only have 6 sections. And we were only given 5, middle of last semester when we were organizing the spring, and I basically begged for a 6th section just because last semester there were a lot of grad students, which justified having more sections. This semester I think there are only 4 grad students, and close to the same number of undergrad students, but I think you're based on the bulletin, you're only allotted, there are 2 4-hour lab sections, and given the numbers, they only wanted to give us 5 total, but I basically asked for 6 and got it. Most of the same lab instructors that you saw last semester. I had to make some cuts, obviously, and hopefully that can change again next fall when you be here. What else? I handed out the lecture schedule in terms of exams. I actually even updated in the manual the lecture topics. So my lecture topics are in the lab manual with the, start with hair, then with what is blood, then all that stuff that we talked about, including the wood lecture midway. And then at the very end, I do a DNA lecture, even though you don't do a DNA lab. And I know there are plenty of you who choose criminalistics because you don't like molecular biology, or bio wasn't your favorite thing. Or maybe you just thought crim was just more interesting, was a better topic to learn about. But I do the DNA lecture, spend quite a bit of time with it, because it's something that, as a criminalist, you're going to be dealing with it. Forensic DNA is actually a subdivision of criminalistics. So even though there are programs that are dedicated to MolBio and forensic molecular biology, we have a track, as you're aware, in forensic molecular biology. As a criminalist, you should know what DNA is. You should know what we do to look for it, and you should know the limits and limits of that particular kind of analysis. So 3 exams. The first one, I won't be here. That'll be just about 4 weeks from now. That's February 21st. I will be in Baltimore for the American Academy of Forensic Sciences meeting. It's kind of late, but I would encourage you. It's not that far away if you have a car or can get on like a train or something like that. Baltimore is not as far away. Last year it was in Seattle. The year before that it was in New Orleans. It's been in Orlando, Florida. It's been in Denver. It's been in different cities in Texas. So it goes all over the place. This is probably the closest it's been in a while, and it's still like 4 or 5 hours away if you're driving. But it's a really great and unique opportunity to learn, to network. This is a huge, really international meeting, so people come from all over the world. and the American Academy has, I don't know, at least 8 to 10 different sections, including criminalistics, things like biology. They have odontology, dentists. They have the medical examiners. They have a jurisprudence section for the lawyers even. So there's a lot of different opportunities to learn. John Jay does have a reception that Wednesday night, but you probably won't be there because you have the exam the next day. So I guess it's really hard to encourage you to be at that meeting if I'm giving you an exam while you're away. Sorry. So the first exam is on the 21st. And just like we did last semester when I was away for the NEEFS meeting, your preference would be to start regular class time at 10:50, right? That way you don't have to, I don't know if you want to sleep late and then, come at 1 o'clock. Okay, so I mean, based on general consensus, I'll probably have one of the lab instructors. I have to confirm it with you guys because I have to confirm to see if Tiffany or Franny or someone else can come and proctor that for you guys, what time they'd be available to do that. But basically, we won't be missing any time then. We've got Thursday off for spring break, and then we'll be done mid-May. Let's see. Here we are on Blackboard. And PowerPoint lectures are there. Tiffany already put in the spring lab manual. I don't know if any of you have the Tuesday section, because I think that's a grad dedicated section with Dr. Reffner, but she did change the time frame of some. Let's see. So these are Tuesday graduate lecture series, open to the public, which includes open to you guys. And there's going to be 4 talks throughout the semester on Tuesdays in the afternoon in this room from 3:45 to 4:45. And I guess I would consider having extra credit opportunities if you show up and maybe write up what you learned in those things. Since I think there are fewer opportunities. Last semester you had moot court. This semester we don't have a moot court. So yeah, even if you have the Tuesday lab section, they've shifted the time frame up on those dates particularly. I believe Tiffany sent out an email already to let you know that on these particular dates, the lab would open a little earlier and close a little earlier, just so that students would be able to go to these particular seminars. And they're, things that Dr. Prinz has put together. You already have Dr. Diachuk over here, our firearms guy, and a number of really world-renowned specialists. So that's one thing. More in course documents. By the time I start talking about blood, after the hair lecture, probably next week, the textbook. Remember you don't need to buy any new textbooks for this semester. You're pretty much using what you got last semester, and whatever's not in that textbook, I'm putting on Blackboard as well as I can. So the Lee chapter text is over here, and this was something I scanned in years ago, because this particular chapter was in the first edition of the Forensic Science Handbook, Volume 1. In the second edition, it was eliminated, and in its place was a forensic DNA chapter, which includes some of the stuff that was in this chapter from Dr. Lee, but it's not as comprehensive. And given that I do both intensive lectures on blood analysis, and I'll probably be spending like 3 to 4 weeks on DNA analysis, you need both of them. I don't know which version of the textbook that you got, either the first edition with the Lee chapter, but the Lee chapter is recaptured over here with a lot of these things that I go over. And a lot of it's the chemistry of biology. And I separated it off. There's a separate, just because this file was huge, 21 megabytes, I made the references in that chapter a second document. What else have we got? Source book in forensic serology. This is pretty old, actually. This is, I think it was compiled by Robert Gainslin, who is also one of the authors of that gray book, or maybe you got the blue book version, basically the book that we use in FOS 313. And he didn't actually write much of the stuff that's in the source book. He really compiled the history of all these different articles that were published. Some of them are even from the 1800s, where people started looking at different kinds of physiological fluids and developing an analytical methods. So it's actually pretty comprehensive, but it's also kind of old, but it doesn't change the fact that, we study these fluids, and these are the original source articles where people began understanding things like, how can we test for blood, how can we test for saliva, how can we test for urine, et cetera, et cetera. So if you were actually to Google source book in forensic serology, you would, there's a link over here for it. But I've also provided on Blackboard source book ID of blood because it specifically talks about a couple of the tests, actually one of the tests, both of them I think, a couple of tests that we use to confirm that a red stain or a reddish-brown stain is really blood and not something else. So I do want you to read this one. It's a lot to ask you to read all of them because the source books are like hundreds and hundreds of pages. It's like a textbook, basically. What else do we have? There is, did I miss it over here? Oh, blood identification. This is an article written by Francis Sheehan. Many of you may have had Professor Sheehan for chemistry. Co-written by Dr. Kobylinski, who was, until about a year ago, the chair of the science department. So this particular reference is going to be really useful for the first few, I guess there's only two, actually three, for the first few blood exercises, because it goes into specific detail about things like, presumptive testing for blood and the different reagents that we'll be using to do color tests. It goes into specific detail about the confirmatory crystal tests that you guys will be mastering in the laboratory. And it goes into specific detail about species identification of blood, because just because you've identified that it is blood, it might be blood from a dog, it might be blood from a cow because you were preparing a steak in the kitchen, or blood from a chicken, same idea. You guys will also be doing a species exercise, species of origin exercise. So this is a really useful reference to look at. But you'll also see that a lot of what's in the Sheehan Kobylinski article is also in the Lee chapter. Okay, so both of those things are available to you. So I've done blood identification, Sheehan and Kobylinski. This is a source book, and should I read the whole thing? Oh, there's a lot in there. Okay, so I will let you know which specific article, because this is huge. There's a lot of pages there. I will let you know which article I want you to specifically read from this particular reference. In the Lee chapter, you can already see some of the blood crystals that you can't really see well there, but you can see these. And the chemical configurations of hemoglobin when you add some of these reagents. And we've already looked at that. So that being the case, do you guys have any questions before I jump into hair analysis? Oh, you're so good. Pay attention. Okay, why am I starting with hair? Well, like I said, this semester we're gonna go from more chemical and physical, chemistry and physics to study of biological materials. And not only that, the other reason why I start with human hair analysis is because you will probably spend a lot of time looking at the hair samples this semester. And so one of the reasons why I start early with this particular lecture is if I actually waited until the end of the semester, because I originally had this as a lecture 6 or 7, it'd be a bit late for you guys. The reason why it'd be a bit late is if you actually waited until, let's say, April to start the hair exercise, you would be so miserable. And the reason why is there are a lot of standard samples to look at. And so you'll be doing a lot of microscopy when it comes to the human hair exercise. The human hair exercise, you guys already did species of origin when you were in FOS 313, and it was designed to be a fairly easy lab because the purpose of that lab was to get you used to chain of custody. Remember, that was the first time that I filled out a chain of custody form in my class, and that sort of thing. And then the hairs that you looked at, if you did the microscopy well, they looked very different. The hairs in this lab do not look very different, but they came from different people. And so it's going to give you an idea of how challenging human hair analysis is. And there's some caveats with this. So as I go through the human hair lecture or the hair analysis lecture, hair specialists or hair examiners do not testify or should not testify to individualization only if all they did was look through the microscope and examine the hairs of interest. Okay? So unless you actually have some DNA information, let's say there was some cells or blood or something like that, the hairs were forcibly pulled out, and you got some DNA from that sample, then you're basically doing DNA analysis to do an individualization, and that would be different. It would just be the source of the DNA was, the root of the hair, typically. Or we'll talk about mitochondrial DNA as well. But the microscopist who specializes in hair analysis, if they don't get any DNA information from it, it's an intelligent hair, there's no cells, then you should not be individualizing that sample. You could do intensive examination and comparisons between the question sample or samples and compare it to hopefully a good representative sampling from the people involved, the victim or victims, the suspect or suspects. And basically looking at the overall information, comparing those two things, the question and known, to make an assessment of the likelihood that the question hair came from a particular source. Okay, but you guys will actually be asked to at least come closer to an individualization because you'll get an unknown for the human hair exercise, and I believe there are at least 10 standard sets, which means 10 people who contributed to the sets, and I think about 15 slides for each set. So you're going to be required to look through at least one full set to get an idea of the range of characteristics in that person's head hairs. A representative sampling would mean if someone came and needed a representative sampling from me, then, and I don't know if they still do this, I have to ask Franny and Tiffany about this, collecting samples of your hair that we'll be using for future standard sets. But a representative sample from me would mean maybe 50, between 50 and 100 hairs. Geez, I hate losing that much. It's getting older, you're losing your hair. But it's the long hairs, thick hairs, short hairs, thin hairs. I've got black hair. I've got plenty of white hair now. I've got some hair that's like kind of brownish. So ideally, if you are collecting a representative sampling from somebody because you need to compare it to an unknown, you want to get examples of all those different things because the question here could be any one of those things. It might be one of the white hairs that came out of my head, and if you looked at me from far away, you might think, oh, I don't know if that, you have to come close and see, yeah, lots of white hair. So you want to get a representative sampling because the question here could have been any one of those things that came off that person's head. And not even just head hair, there's pubic hair, of course, as well, and other body hair. Although typically, we're mostly concerned with head hair, which falls off naturally, and pubic hair more likely if it's some kind of sexual assault situation, but not necessarily. Of course, there are going to be hairs, with someone plucking their eyebrows, or did an eyelash fall out, or is it an underarm hair, is it a leg hair, an arm hair? And for those things, the hairs from different somatic origins, where in the body did it come from? It's not that they won't be relevant. The question is, like, what kinds, actually, the question is, what's the question? Like, are we trying to determine what kind of hair this is? Possibly. And along the way, though, the more pressing question is typically, who did this hair come from? Okay? Because if you can make those connections, this is a pubic hair, and it clearly did not come from the victim. If you examine the victim's pubic hairs, etc., and it was a sexual assault, now you want to make some comparisons with, let's say, perhaps a suspect that was arrested to see if it was consistent with that person's pubic hair. So those are the kinds of things that we look at. But then, there might be, like I said, there are hairs all over people's bodies. The only place you don't have hair is like the palms of your hand and the soles of your feet. You still have peach fuzz on your face, and the hair is basically all over the place. But when is that going to be relevant? When are you going to find those and say, oh, that's really a key piece of evidence to look at? It'll depend on the situation. So I'm jumping ahead a little with this, but like I said, the reason why I would encourage you to start the hair exercise early is because I'm not expecting you to finish it early. What I'm advising you is, why don't you spend maybe an hour looking at some hairs and documenting them, and then when your eyes feel kind of sore from staring through a microscope for that long, then move on to a different exercise. And so what I'm saying is revisit the hair exercise throughout the semesters to familiarize yourself with examining the different properties of human hairs through the microscope using the PLM. And then you'll eventually get your unknown at some point in the semester. But you want to, like I said, if you save all of that for the end of the semester, it's going to be a real headache looking at all of those hairs for hours on end, 4 hours straight. That's really a lot to ask of a human being. You're going to get a headache probably just from staring at hairs for so long. The hair exercise a couple of years ago, like I said, even though in the real world, I would never expect you should not ever testify that I examined the human hair under the microscope and I've individualized it to have come from this person. Maybe if you did the DNA, you could testify to an individualization, but using microscopy alone, the best you should be able to do is indicate that the hair is consistent. It could have come from this person, which is very different from saying it must have come from this person and nobody else in the world. So that's a real world situation. I think I may have mentioned to you maybe in the last, in 313 or last semester, a few years ago, the FBI actually did a review, and they decided that they would review the hair testimony of all their hair examiners. And why was that? It's because their hair people were testifying beyond what science could justify. So basically, some of these hair experts were testifying in a court of law and trying to help the prosecution. And so they would basically be asked questions like, what's the likelihood that that hair came from Mr. Defendant, for instance? And they would basically pull numbers out of their ass and say, oh, you would see that, maybe 1 in 100 times, or 50 out of, they would try to use a number to connect the question hair with the known hair with no scientific foundation to that numbering, to those statistics. And because several of their experts were testifying beyond what science could justify, the FBI had to kind of review the testimony of all their hair examiners. That's a lot. If you think about it, they're going back, let's say, maybe at least a decade of what people testified to, and the possibility that, okay, if it was the only evidence, then it probably was something that was used to, basically deem a person guilty of a particular crime. And other times it might have been one piece of evidence along with many other things, and the court might decide it was harmless error for that testimony, even though it it was not justified. There were so many other things that would have given the same verdict anyway. But why are we asking you? Well, like I said, up until a couple of years ago, we were asking students to individualize. When you do your human hair exercise, if you feel pretty confident that your question here came from this one set, then you can put that in your report. If you're wrong, and then you know how it goes with getting an exercise wrong, you know, 0. You could request to do it again, although a lot of people say there's no freaking way I'm going to do this again. And some people say, okay, I'll do it again. But up in, a couple of years ago, we decided that because it was, you know, people should not be individualizing when they testify about hairs. You have the option, if you can't decide between, let's say, my unknown and set A and set B, then you can testify. You could write in your report that your questioned hair has characteristics consistent with both sets A and sets B. But you'll also be starting off with a lower factor. But it's a little bit more realistic. And like I said, the other part of this is that it's a closed set. Your unknown hair could only have come from one of those sets that's in the laboratory. In a real world situation, the unknown hair could have come from anybody. Okay, so of course there's a much larger set to be concerned with, whereas in the crim lab, it's a much smaller set to be concerned with. So there's, you know, just different nuances with that. So that's the reason why I would encourage you to start the human hair exercise fairly early. Okay. All right. This should look familiar because, like I said, probably 99% of the slides over here. I've only added a couple of extra things for the senior level with hair analysis. So like I said, I'm going to go through this fairly quickly because it should be a review of what we did in FOS 313. Okay. Microscopical analysis based on class characteristics. You know, what color is it? What are, and we'll talk about these other things like the modulation. What does a root look like? What does the pigmentation look like? Is it changing from root to tip? Because that's the other aspect of it. Bear in mind that when you looked at the hairs in FOS 313, most of them are pretty short. Didn't take too long to go from root to tip with those things. Some of these hairs will be a bit longer, and you should look at from root to tip for every single one of the hairs that you're analyzing, okay? Because the properties can change. My own example is I'll have some hairs that start off, you know, blackish or brownish, and then they end up being white after a little while. Same hair, okay? So things will change as you go from root to tip, and you want to make note of those kinds of characteristics if there's some changing along the way. And some comparison to evaluate the commonality of origin, but like I said, we do not testify to individualization when it comes to hair, okay, using microscopy alone. If it's DNA, then maybe we could say something about it. And some of your friends who are in the toxicology track of the forensic science program, they'll probably learn more about things like, you know, drug analysis when the biological sample that's been obtained is hair. And many of you, when you start applying for jobs, a lot of the jobs are civil service jobs, which means you'll be working for the government. It won't be unusual that as part of a pretty stringent application process, you'll be asked to do things like a psychological examination. You'll be asked to do a lie detector test, a polygraph. You might be asked to do a number of those things. Plus, you may be asked for a urine sample, or you may be asked to come in so they can get some hairs off of you, and typically for the purpose of analyzing to see if you've been using controlled substances. Okay. And the basic idea of this would be somebody, let's say, is smoking marijuana on a regular basis. Not naming names or pointing at anybody. Those metabolites, tetrahydrocannabinol and cannabidiol, they're going to end up in your system because you're metabolizing them. And some of those things, some of those biochemicals may end up in the follicle as the hair is being produced, and those metabolites get incorporated. So one of the reasons why they like human hair analysis for drug analysis is you basically get a timeline. So from root to tip, at the root is the most current situation, and as you move along, typically the average is people grow hair about 1 centimeter per month. So if you've got like a 10 centimeter length hair, then you've got 10 months of history for that person. And what they will typically do is, if they have a hair like that, they will cut it into 1 centimeter lengths, labeled based on which one's closest to the root and which one's furthest away from the root. And they will analyze each of those segments to get an idea of the timeline if this person's been using a controlled substance. And obviously it could be anything like cocaine, heroin, fentanyl, methamphetamines, any of those different things might be incorporated.
How does that change for someone who's like bald, or they have their hair cut short like this,
They don't have to just get head hair.
But like if you're a trimmed person or a clean person, how could they trace back that far?
Well, they can't. Not from the hair.
Oh.
Yeah. So if you've got really short hair, then well, like I said, they might choose to get hair from a different part of your body. But even that is, most people don't have really long hair elsewhere. They may do a combination of a urine test, and like I said, they could look at your hair as well. And bear in mind, it's not just necessarily in the application process. Let's say you've been working, I think this is like an issue, let's say you are the engineer on the subway train, right? I think there was an accident several years ago where they were concerned about, was the guy operating the train, was he drunk? Did he fall asleep? Was he on drugs? And that sort of thing, because there was a train accident.
The Metro North? The Metro North thing?
I think Union Station.
There have been Metro North. There's been a couple of problems, several times actually. There have been problems, like with zooming along and trying to get on curve at a really high speed, and the train derailed. So you do have situations where they might look at the engineer of the train to see if were they taking drugs. And so this is already an employee that they want to check to see if there's a history of drug use that might have affected their ability to do their job well, because people died, that kind of thing. So yeah, and so it'll depend. Like I've had people this is more like when I used to teach FOS 108, and I'm going in for the police exam, is it worth it for me to shave my hair? And I'm like, don't talk to me. I don't want to talk to you. You're asking me about how you can get away with, because you've been enjoying college a little bit too much. Do these shampoos work? Or, no.
Eating
Talk
bagels
about it.
and stuff.
But yeah, as part of your application process, like I said, I think many of you who, when you finish, probably in May or pretty close to that, you're going to be applying for jobs, and these are the things you're going to be encountering. Like I said, a lot of places will do the urine test, but some places will pluck a hair from you or a couple of hairs to see what they get, and that's part of the situation here. So that's the basic, metabolism of a drug, and it ends up incorporated into the hair, just like your mitochondrial DNA is also incorporated into your hair. So those are a variety of things that they could look at. And I don't want to discount, actually, as I go off on another tangent, the possibility that the surface of the hair has information as well, like maybe residues from the shampoo you used, or you live or work or you smoke, and there's a smoky environment. There's going to be residues of these chemicals in your hair that people don't always look for, but if you think about it, it's trace evidence. And it's something to consider when you're examining a hair. So rather than, we get the hair, let's wash it, and then examine it, put it in the microscope slide and examine it, there might be compelling reasons for you to think about, well, is there a reason to look at the chemicals, maybe swab the outer surface of the hair before you prepare it for microscopy, that kind of thing. Trace evidence, talk about that. And another tangent along the way. At the end of the semester, you guys will be doing a trace evidence exercise, which is basically like a capstone exercise for the course. I believe that Tiffany put in the manual at the last date that that you have to hand in. All the other exercises, like early in May, maybe May 7th, I can't check the date yourself. After that, everyone should be focused on the trace evidence exercise. And what my lab staff does is they generate items of clothing typically, might be shoes, socks, jeans, underwear. Sometimes it's like a purse. Haven't seen that in a while though. And it's part of a scenario. The scenario is already in the lab manual. Typically there's 2 victims, 2 suspects, and you will receive an item of clothing from one of those people, and you will examine that item of clothing and look for trace evidence. Are there stains? Are there fibers? Are there hairs? Are there any other kinds of things of interest? And then in addition to that, a few things. It's a very involved exercise. You have to make decisions about prioritization. Like if you found some fibers, maybe you found some glass, but you also found a stain, which pieces of evidence will give you, will be able to answer the most relevant questions for that particular crime. Remember, you want to try and make connections. Is this a fiber that on the suspect's jeans that could have come from a victim's sweater? But is this blood on the suspect's t-shirt? Could it have come from one of the victims? Now, you guys aren't going to be doing DNA analysis, but you want to consider the idea that if you've determined that it really is blood, that this is a really important piece of evidence, and I would probably forward it to the molecular biology lab to get a DNA, to attempt to get a DNA profile on that. So that's probably one of the more important types of evidence that's higher on your priority list compared to the fibers or the glass, which are not individualizable, but useful because they can help with connections. So you have to make decisions about, and believe me, my staff does not put like 200 trace evidence pieces on the jeans you get, but they're, someone's clothing, and, sometimes if you're very zealous, you can find, I've seen people with lists of like 100 things that they found on, the item of clothing they received. Some of it's just incidental, okay? So part of it's prioritization. And then, let's come back to that glass with the fiber. You have a pair of blue jeans and there's a red wool fiber, and then you look on the list of different items and you see that, victim number 2 had a red wool sweater. So let me find out which of my classmates was assigned that particular piece of evidence and request a standard that came from that so I can do a comparison, okay? What will that comparison tell you? Well, maybe it's, maybe it's not that one of them is not wool, one of them is like a polyester, one of them is wool, then you've excluded, okay? Or if you find that they are the same type and they have similar characteristics, then you've included the 2 fibers as possibly coming from the same source, but fibers are not individualizable. So you're not going to say this fiber must have come from this victim, but you could say this fiber could have come from this victim, which is still useful. So you will be collaborating with your classmates in solving this crime, okay? And it's a culmination of things. When I did this exercise when I was like a wee little grad student many, many years ago, in 1990, this would have been 1990, spring of 94, I got a pair of jeans from a suspect, and when I was analyzing it, you have fresh bench paper, you'll have your fiber optic light sources, you'll have the stereo microscope, typically hopefully on a boom so you can move it around to look at the evidence, or look at the evidence and move it around if you can't move the microscope as much. And I remember I looked in the pocket, one of the pockets of the jeans had what looked like poppy seeds to me. I'd never seen this before. I was like, wow, why the guy have poppy seeds in his pocket? And as I analyzed these things a little more, it turned out to be gunpowder that he had in his pocket. It looked a lot like poppy seeds. Gunpowder actually has a lot of different possible formulations. Some of it looks like round balls, like poppy seeds. Some of them look like discs or flakes. Some of them look like little cylinders with a hole through them. So it depends on who makes the gunpowder. But the stuff that I happen to have, so you might encounter some evidence that you have never worked with before. And that's another thing to think about, okay, in terms to how you're going to approach that analysis and make decisions about prioritizing it with the other kinds of trace evidence that you discover. So DNA analysis, nuclear, if you have cells and blood at the root, and mitochondrial DNA more often, but it depends on the situation. People who have very light-colored hair have less mitochondrial DNA in the length of their hair. People who have darker colored hair, like me, will have a lot more mitochondrial DNA in the length of their hair, so you're more likely to get a mito profile from my hair than you would from someone who is naturally blonde and doesn't have as much mitochondrial DNA in their hair. So those are some situations to consider. So mitochondrial DNA is in the shaft of the hair to different quantities for different people. Nuclear DNA, you're not going to find it unless the hair was forcibly pulled and you have some skin cells and or blood attached to the root of the hair. Okay. So with the nuclear, if you do get that, you might be able to get a full CODIS profile, a full DNA profile, which is something that can individualize the sample to the person it came from. The mitochondrial DNA came from your mom, which means that you have the exact same mitochondrial DNA profile as your mother, as any of your siblings with the same mother. The only time it isn't the same is if someone mutated along the way. Okay, and even then the mutation is typically maybe one base or something like that in the mitochondrial DNA. Hair's common, easily falls out and sticks to many surfaces. We can look at those characteristics. The hair is on the surface of the passenger seat of the car. So, yes?
Can you just repeat where you said in the hair, the mitochondrial DNA?
The mitochondrial DNA, basically, mitochondrial DNA is incorporated into the hair much the same way as your pigment granules are. But your mitochondrial DNA is very different from your nuclear DNA. Nuclear DNA is in the nucleus. Mitochondrial DNA is in the mitochondria. So I don't know if you remember from biology, if you see a picture of a cell. And I will do this, I will go over this more specifically when I do the DNA lecture at the end of the semester. You've got your cell membrane, you've got that big round thing, that's your nucleus, and it's got basically one copy of all 23 pairs or 46 chromosomes. And then you might have a bunch of these little, much smaller cell organelles, which are mitochondria. And the mitochondria have their own circular DNA. So that is distinct from the DNA that's inside the nucleus. The nuclear DNA, that's a combination from your mom, half from dad. The mitochondrial DNA is just from mom. Okay, so just from mom means it's not individualizing. It'll be the exact same sequence as what your mom has and what your siblings have and what your grandma on your mom's side and basically everyone in the maternal line. But if you're the guy and you have children, your children will not have the same mitochondrial DNA profile as you do. They'll have the mitochondrial DNA profile of the mother of the child, not the father of the child. So those are things that we look at. And just as an aside, we look at mitochondrial DNA to look at basically the history of female DNA, whereas we look at Y chromosomes to look at the history of male DNA. When I talk about history, I'm talking about ancestry, and this is how some biologists go back and say how many original people were there to the original Adams and Eves that produced all of us, and we're all related in some way, et cetera, that kind of thing. And then we'll look at the sequences in those things to see how people have differentiated from each other in the female line using mitochondrial DNA, and the male line by looking at Y chromosomes. It's one of the ways that they figured out, was it Genghis Khan, one of those conquerors in Asia, went and with his armies ravaged various areas in Asia, and apparently made a lot of children too, because his Y chromosome is in a lot of men. So that's one of the things that they were able to figure out by looking at Y chromosomes, that all these people had a common father or grandfather, great-great-great-great-great-great-grandfather along the way. So those are some of the things they look at. To answer your question. So you can figure out things like, was the hair freshly deposited because it's on the top surface of a particular item? Or was it there a long time because it's like ground into the fibers of the carpet, and you have to like, when you try to extract the hair, it's been there a while, okay? Or it's very dusty and dirty. It's probably been there a while. How long? Can't really put, you can't really date the time that the hair was falling out and on that surface easily. But you can provide estimates. Just like the idea that is it a fresh blood stain or an old blood stain? Well, fresher blood is going to be more of a reddish color. An older blood stain will look like a brown color. But you might have a blood stain that turns brown very quickly because it's very hot, it's very dry, so it depends on the environment. Or it might be a blood stain that still looks fairly reddish because it was a very cold environment, for instance. So you can't make specific, you have to be careful about suggesting how long that stain was there or how long that hair was there. Or other aspects like that hair looks like it was physically ripped out because you got all these, like you got blood, and these are anagen hairs. These are hairs that were actively growing. You got blood and cells at the roots, and there probably are several of them. So these can help you provide information for the context of that hair. Context meaning, what situation put the hair in that condition in the place that you found it. Okay. And microscopy and DNA, complementary, meaning that now that we have great DNA analytical methods, it doesn't mean toss out the microscopy. There have been, I've worked on cases where the investigators pick up like more than 100 hairs from the crime scene, and you certainly do not want to send all those 100 hairs to the DNA lab. DNA laboratory analysis is really expensive and time-consuming. Makes a lot more sense to have the microscopist kind of sort through those hairs, examine them. This set looks like it came from the same person. This set came from a different person, and then just pick a few of them. Maybe you could see if there's a root that's got some cells on it. That would be pretty useful to send for analysis in the DNA lab. So instead of sending 100 hairs and spending tens of thousands of dollars to analyze them, send a choice few based on microscopy. Okay, so these methods complement each other. They provide useful information that together is synergistically more informative.
So, like, what kinds of things would a microscopist look for? And say in the case that they did have 150 hairs, like, and I don't know if most of them normally have the roots intact when they're found. I don't know how this goes.
Well, when you say roots intact, there would be the difference between was the hair cut,
yeah,
or was it
pulled off.
a root that just naturally came off. I'll talk about those growth stages, right?
Yeah. So like,
well, let's say you didn't vacuum your house in a few weeks, and so there were a lot of hairs that were on, in the place, right? And then something happened, and they started collecting hairs from the crime scene, right? So you potentially have a lot of hairs, and a lot of those hairs might be from people who happen to live there that have nothing to do with the situation of concern. So what was your question with respect to what the microscopist does?
yeah, how exactly would they read them out? Like, say like half of them had roots. Like, would they consider the location that they were found to?
They should be. Yeah, they should.
Oh, okay.
Yeah. You want, I guess, I will
How would you
allude to
exclude
this
those?
throughout the semester, but if you haven't kind of gotten the gist of, what I talk about, the context of the evidence is really relevant, which means where did it come from? Where was it situated? What else was surrounding it? Can help to either answer questions or pose more useful questions in terms of how should I analyze this particular piece of evidence. Okay, so when you get a piece of evidence in isolation, someone in the investigative team in the laboratory should have more information about its context. A lot of people fear that with too much context information when you're analyzing a piece of evidence, you're going to use your bias, and your bias will affect how you interpret the results. So one of the suggestions that's been made to make sure that context is incorporated, but to minimize the bias, it makes more sense, let's say, for the supervisor to look at the evidence, to consider the context, pose the questions, and design the analysis, and then have a technical person do the analysis, and the technical person doesn't need to know all the context information, but someone does. So if the technical person can do the analysis, minimize bias, and then provide the results to the person who does know the context to see how it all fits in. Okay? But it makes a lot more sense than just, okay, we plucked this thing, I'm not going to tell you where it came from, I'm not going to tell you anything about it. But then in those situations, what typically ends up happening is either a police officer or a lawyer is telling you what to do scientifically, which has a lot of problems because that person doesn't know all the science that's involved, doesn't know necessarily what the capabilities of the lab are to interpret different kinds of evidence. So they're telling you to do what they think you could do, and sometimes it's missing the point. Sometimes you're answering a question that is not really useful or relevant, and it's better to have at least some kind of communication between those two things. It's one of the things that when you go to some of these conferences, especially Dr. DeForest's talks, he talks about how a lot of the laboratories, not all of them, but a lot of the laboratories are moving towards this clinical lab approach where it's the problem that I just talked about, that someone basically says, here's a sample, I want you to do X or Y or X and Y analysis on it, and just give me the results, and there's no context, and you're basically just reacting to what someone else has told you to do, as opposed to a better situation where if you're trying to investigate a crime, for instance, there should be a dialogue between the scientist and the investigators that procure the evidence to determine what's the context, what's the background information, what are the useful questions to ask with regard to this evidence and how it related to the crime, and therefore, okay, well, we could do this in the lab as well, and having there be a dialogue so that it's not just, I'm going to react to whatever you tell me to do, versus I can provide useful information to you from a scientific perspective, and there are additional tests that could be done that you probably didn't even know about because you're the lawyer or you're the cop or whatever the case may be. So that's my little podium that I step on based on what I've learned myself in doing casework and that sort of thing. Where if you allow yourself to be just told what to do, sometimes the point is missed. That said, most of you are not going to be in a position when you start working in the lab to start, telling people what to do and that sort of thing. But it's something to keep in mind moving forward in your career. For the most part, most of you will start off in that technical role, where let's say your supervisor knows a lot more about the case at hand, and you're given the evidence to analyze. And then hopefully at some point, you move up the ladder in your career, eventually supervising others, eventually working more with investigators and lawyers, and hopefully having a good communication with them to be able to, bring out the important questions. So, this you already know, the hair is produced in follicles. It looks like it goes pretty deep down, right? Meanwhile, what is not characterized, well, some of your hairs pop straight out, but like if you think about your eyebrows, the follicle is actually angled in, right? So depending on the part of the body, this right angle of skin and follicle is not necessarily always what you have biologically. But you have your follicle, which is basically like this miniature organ that you have all over the place, like on your skin, and your circulatory system has capillaries that feed into the follicle, and that has to make sense because what's your hair made out of? Protein. Where's it going to get the protein from? From your blood, from your blood system. So your blood is circulating, it'll bring proteins in over here, it'll bring metabolites from what you've eaten, or if someone's snuffed cocaine, et cetera, some of that will show up in here. The bottom portion is kind of squishy, and the hair does not get hardened or keratinized until a little bit further up. So if you pull out your hair while it's still actively growing, you'll notice that at the root end, it'll be very soft. Okay. Hair is fairly robust, both physically and chemically. Physically, I think I've mentioned before, you could take it and rip it in half if you want. You could step on it and grind it under your feet. But in either of those cases, you could examine it under a microscope and say, that's a hair, okay? And that's the more realistic point of having it have physical, strong physical structure. It could be hot, cold, rainy, humid, dry. Someone could step on it, but when you examine it, it's still a hair. It's not going to dissolve over time. In fact, if you exhume a body, a lot of the times, if it's been several years, you might find skeletal remains, and you might find hair as well, because the hair will stick around more. It'll last. Okay, it's resistant to environmental decomposition, and it sheds very easily. With the chemical structure, long strands of keratin cross-linked with disulfide bonds. So the keratin is an insoluble and chemically resistant protein. Chemically resistant means that you need some fairly harsh chemicals to have a chemical reaction occur, and you don't normally have those kinds of harsh chemicals in an ordinary environment. You've got to go to the hair salon if you want some harsh chemicals to either straighten your hair or get a permanent curl or wave, or to dye the hair in a particular way. And like I said, you could put hair in water; it's not going to dissolve. Okay. Major constituents, keratin, not only hair, but also your fingernails and your toenails. In birds, their beaks, their feathers. In other animals, their hooves, their horns. Lots of these things, they're all keratin. And what's providing our disulfide bonds? It's the sulfur in the cysteine, which you learned about in biochemistry as one of the several amino acids. Cysteine, its group is the S and the H, sulfur and hydrogen. If you put two of them together and basically dehydrate it, you get rid of the hydrogens and the two sulfurs link. They bond together, which is a pretty strong bond. That's your disulfide bond. When they cross-link, two cysteines becomes cysteine. So you basically take out the E to show that it's two of these things. So that's cysteine over here. And then when two of them combine, your SH group, you get rid of the hydrogens. And the sulfur from one cysteine bonds to the other cysteine. And you have a disulfide bond. So when you have a protein, it'll have cysteine along its length because a protein is a chain of amino acids. And in places where you have a cysteine on one protein strand and a cysteine on another protein strand adjoining it, you have the potential to have this cross-linking occur. And this is one of the things that makes the hair strand particularly chemically strong, because without the cysteine cross-links or the cysteines that you have, if you want to think of the protein strands as being these long, thin chains like threads, then one thread can easily move along the next threads near it. If instead you have these disulfide bonds, now those threads or those protein strands cannot move alongside each other. They're kind of stuck where they are because they're bonded one protein strand to the next protein strand because of those disulfide bonds. And that's what you see over here. So the green is supposed to symbolize the strand of protein, which means lots of amino acids all bonded together in a long chain, which that's what makes a protein. In the places where you have the sulfur sticking out, that happens to be where a cysteine is in the protein strand. There's one over here and one over there, and you've got your cysteine, where you have a cross-linking occurring over here, over here, over here, which means that these two strands can no longer freely move and slide against each other and change positioning, okay? They're stuck where they are, and once again, this adds to the chemical stability of hair, okay? For perms and straightening, like I said, you've got to go to the hair salon to expose your hair to thioglycolic acid. This will reduce the sulfur bonds, so it'll break them, okay? Once those sulfur bonds are broken, You can either have the hairstylist put your hair in some kind of curler, or put your hair to something that'll straighten it out, and then they will apply another chemical which will reform these disulfide bonds, that's the hydrogen peroxide, and the idea would be the cysteines will have shifted, and when they reform, the cysteines are bonding with a different cysteine along the protein strand, and that's why it's called a perm, permanent, because it's the bonding which is going to stay in place after you've done this harsh chemical treatment of the hair. And that's why the permanent wave, the hair will have that wave after a while. Or if you're going for straightening, the same idea. You've broken the sulfur bonds, you've shifted the positioning of the cysteines along the protein strands, and then you reform different cysteine bonds. Depilatories, basically those are hair removers, and in this particular case, while hair is stable in both acidic and alkaline pHs, if you first break the disulfide linkages, which means, let's say you expose the hair to thioglycolic acid, breaks those bonds, and now you put it in even a slightly alkaline or basic pH, the hair can come out more easily, actually. And that's what depilatories do, chemically remove the hair instead of shaving. So the thioglycolic acid is for breaking those disulfide bonds, and the calcium hydroxide is for the basic pH. Structure of hair, this is old, 3 layers, the innermost layer, the medulla, the outermost layer is the cuticle, and the one that's in between, which for humans is the thickest layer, is the cortex. The scales on the outside overlap like roof shingles, so that even though you see this line over here, it's the end of one scale, at the other part of it, it's underneath the scale that's next to it. Okay, so only a small portion of the scale is exposed, the rest of it's underneath another scale. Okay, so if you, just like the way fish scales are or roof shingles are, you put one over the other, and if you were to put water, it would be harder for the water to get inside because you've got these overlaps. Okay. The average thickness of a cuticle is about 6 layers of scales. So that gives you insight into how long these things go back into the cuticle. Okay. And they point away from the root, which means that you can feel the friction if you take a single hair and run your finger from tip to root, you'll feel more friction resistance because you're going against the scales, whereas you should feel less of that if you're going from root to tip. Okay. And in terms of healthy hair, the whole idea being the cuticle is your protective layer. It's like the armor on the outside. The scales are like armor on the outside of the hair. So if the hair gets damaged from heat, from chemicals, the scales might come outwards instead of staying against the shaft, and then you start getting those things like split ends and that sort of thing, which is damage to the hair. So the cuticle is very inert. So like I said, it's like your armor. Doesn't chemically react easily. Lots of sulfur in there. And different animals have different morphologies of the scales. Okay. The cortex, so we're going now the next layer in, which is the thickest layer for humans. But you already saw in the animal hairs, you saw in FOS 313, the cortex is very thin for other animal hairs. Like the deer hair, remember the deer hair with that cobblestone path? That's all medulla, and it took up most of the shaft of the hair. So that meant that the deer hair has very little cortex. It has a lot more medulla, and then it's got the cuticle on the outside. Okay. The cortex contains tightly interlocked spindle shape. Spindle shape means thicker in the middle, tapers off at the ends. Spindle-shaped cortical cells, about 5 microns in diameter. And you can see a little bit of what's already alluded to over here with an individual protein strand that's depicted as a line. If you bundle a few of those together, like 2 or 3 of them, you get a protofibril, and those are also disulfide linked. If you bundle a few of the protofibrils, you get a microfibril, and if you bundle a few of those, you get a macrofibril. And so it's showing you the exact same thing over here, where it's not even showing you the individual strand on its own. You've got a protofibril, which is like 2 to 3 protein strands, and then that's bundled. And so basically, the structure of the proteins are, it adds to the physical strength of the hair. Okay, so it's harder to pull it apart because it's basically like taking a very thin thread. It's going to be pretty weak and easy to rip into two. But if you take 2 or 3 of them and kind of twist them all together, and then you take a bunch of those and twist them together, then you take a bunch of those and twist them together, and the next thing you know, you have a big, thick rope. And that's going to be much harder to pull apart. Okay. So the macro, we'll start from the small. The protofibrils are 2 to 3 interlinked or intertwined cross-linked alpha helices of protein. And a protofibril is about 20 angstroms, which would be about 200 nanometers in thickness. Okay. Microfibril is about 4 of these things. 20 times 4 is 80. And then the macrofibril, you've got a few more of these bundled together, and it's basically the same idea as what you see over here. Okay. The cortex contains your pigment granules. Pigment granules come from the melanocytes that are in your follicle. Melanocytes are pigment-producing cells. They're embryologically related to neural cells, and basically the melanocytes incorporate these little melanosomes, those little round specks of colored material. Okay. Melanosomes are also called pigment granules. Those are interchangeable terms. And because they're similar to mitochondria, it's related to that idea of what I told you before. The more pigment granules a person has in their hair, the more mitochondrial DNA, because it's basically all those things are being injected into the hair in the follicle as it's being produced. Okay. So melanin is basically the name of the pigment in melanosome. It's actually a family of pigments. It's a polymer of indole 5,6-quinone. And there are 2 main types. There's eumelanin and there's pheomelanin. Okay. Eumelanin, you've got 1 and 2. Monomer 1 is the black pigment. So you'll see that more in people of Asian origin or African origin. Their hair has a lot of eumelanin monomer 1. But it might also be mixed with some eumelanin monomer 2, which is some brown pigment. Okay, so I've got a little bit of both. Pheomelanin is a yellow to reddish brown pigment. So if you have red hair, you have no eumelanin, you only have pheomelanin. Okay, if you have blonde hair, you may have a little bit of eumelanin, the brown pigment, but a lot more of the pheomelanin. How is that decided? It's genetic, based on your parents. And hair color is not based on a single gene. There are several genes which are used, and we don't fully understand it all. There are companies who will take a DNA sample, a biological material, they get the DNA out of it, and they will examine it, and they can come up with what the hair color, what the genetically, what the color might be. Okay, so it's one of the things that they can look at. So let's say if they have like a seminal fluid stain, not a hair, they can examine the seminal fluid for the nuclear DNA, and they might be able to say, okay, the suspect has red hair because there's no eumelanin. Okay, that kind of thing, to help with getting some kind of physical profile of the person. So that's chemistry. And what's wrong with this picture? I think I showed you this like in FOS 313, right? Do you remember, or did I? Maybe not. Like I said, I threw in a few more slides over here. Where's the pigment granules in this picture over here? They threw them right in the center. What's in the center of the hair? Medulla. Medulla does not contain pigment granules. Pigment granules are in the cortex. Okay. That's what happens when you get these like henna for hair websites where they make these kind of scientific errors. The cortex will also have things like cortical foci, which are these tiny little air bubbles. They tend to be small, and when you look at these with the hairs that are in the hair exercise, Some of the hairs will have cortical fusi, not just in the root area, but some of them might have them throughout the hair. It's one of the characteristics that you look at. One of the many reasons why human hair analysis is complicated is because as you look at the many different hairs from a single person, you'll see the characteristics in one hair is shifting or changing from root to tip. And if you compare it with another hair from the same person, those characteristics are not changing in the exact same way. So you're looking at basically a compilation of all these characteristics and how they might be different for a single person, and it might not be too strange then that another person might have a hair that's very similar to that, okay? Which is why we don't individualize human hairs. Ovoid bodies are, I need to take out that question mark. Basically, an ovoid body is like a clump of pigment granules. When you compare a cortical fusi, an air bubble, to a clump of pigment granules, you'll see that as you focus through that little circle, you'll see something different. So that's useful in distinguishing a cortical fusi from an ovoid body. Basically, if it's an ovoid body, then it's a clump of melanosomes, and as you change the focus through the different part of that little structure, it'll remain dark the whole time, okay? Because all you're doing is looking at a different layer of focus for a clump of pigment granules. If, on the other hand, it's an air bubble, then you already know that there's air on the inside but not on the outside, and an air bubble will go from being like light to dark to light. So that'll be the difference between what you see for a cortical fusi and a pigment and an ovoid body. Okay. So here are some cortical fusi, and you can see as I switch from these to the ovoid bodies, you might say they look kind of alike. So a single picture is not going to necessarily tell you the information you need. You need to focus through. And by focusing through, I mean changing the height of the stage. So you're looking at different parts of that little circular structure to see what it looks like a little bit deeper in and on the bottom and the top. But yeah, they look like dark things. You'll also see in these particular samples, over here, you've got your medulation, which means you've got air in the dark areas over here, but some places where there's some cells inside the medulla, you've got a fairly light and uniform distribution of pigment throughout the cortex over here. You can see the cuticle on this side. If you, there are a couple of ways where if you've got really good, you're tight with how you've controlled the illumination, you can actually even see the scales one on top of each other. But you can also use polarized light microscopy and your quarter wave compensator to help you visualize that well as well. Okay, so you can see some differences over here. Whereas over here, much darker pigmentation, and it's more streaky. Sometimes you'll see pigment that seems over here. It seems like the pigment's a little bit closer to the medulla and less of it closer to the cuticle in this area, but over here it's a little bit more distributed along the width of the hair. And you can even see to some extent that this particular medulla is not dark. So there's cells inside the medulla over there. But you can see it looks a little bit lighter over here, right? Because those cells have a different, basically a different refractive index, and there's no pigment inside of the medulla. And that's why it looks a little bit lighter over there. Okay, so you can see the medulation even if there's no air in there, and it's not patterned like you have for certain animals. Okay. Oh, and over here, once again, some streaky and fairly well distributed pigmentation, but you've got like a ball of pigmentation right over there, and you can see that that's just something that that person happens to have, maybe genetically part of how their hair is. So every now and then, just like a big clump of melanosomes all stuck together. And remember, I frequently use this as an analogy for resolution. This ovoid body is consisting of many small melanosomes all stuck together. And you haven't resolved the different melanosomes because they're all stuck together. And the other analogy I give you is like, you're looking at the traffic light, and it's really composed of all those different little smaller red lights. Far away, it looks like a red circle. Closer up, you can see that it's actually made up of a lot of little red dots. So that would be the traffic light equivalent to looking at an ovoid body, and you have not resolved the individual melanosomes. Okay. So for the medulla, central canal, the cells are amorphous. Remember, the cortical cells are, they have that thicker in the middle and tapered at the end. Also sulfur-rich when you do have those cells. Sometimes you don't. If there are no cells inside that canal, then it's air, and if it's air, it's basically the reason why it looks dark is total internal reflection. That again. I keep talking about total internal reflection. Total internal reflection, you guys, when you used your Abbey refractometer, it used total internal reflection at the critical angle to determine the refractive index of the liquid. And the reason why the hair will look dark when there's air inside is because the air has a much lower refractive index compared to the cells surrounding it. So that's why you have total internal reflection. It's sort of like what you have with fiber optics, those long fibers where there's total internal reflection inside of fiber optic. And you already know about it. You've seen a few of those by examining hairs in FOS 313, but there are far more that you haven't looked at. Hair has ion exchange properties and will take up and bind certain ions like arsenic. But that also means that there's a difference between someone's poisoning the person and they put arsenic in their food and they've ingested arsenic and they're being poisoned. And so arsenic in their hair coming from their diet versus take the hair and soak it in a solution that contains arsenic ions, and the arsenic ions will incorporate themselves into the hair. So you need to be careful about trying to get an understanding of where did the arsenic come from. Did it come from the person's diet, or did it come from having it, exposed to arsenic in the environment? And you might think, well, most people aren't using arsenic shampoos, so they're not going to have arsenic coming from an outside source. This is more the situation where you've exhumed a body. Let's say, they thought they didn't detect arsenic when the person originally died, and maybe the medical examiner said maybe this was natural causes. And then there's some question that maybe the person was poisoned with arsenic, and so they may exhume a body, and they will look to see if they can detect arsenic in the remains that are left behind. And one of the things that they need to be careful of is if there's arsenic in the soil, in the surrounding place where the person was buried, they have to collect some of that soil to determine whether or not there's a possibility that some of the arsenic may have come from that environment in which the body was buried. Okay? Because you can't distinguish intrinsic from their diet, arsenic consumed, or arsenic that was incorporated from an external environmental source. Okay. Hence the failure of neutron activation analysis. Back in the 50s and 60s, they used this. Did you guys even learn about neutron activation analysis and instrumental analysis? Mm-hmm. No, they don't talk about it, right? Mm-hmm. Okay, because it's not that it's a method that's no longer used. It is a method that it's just a difficult method because neutron activation analysis requires basically the equivalent of a nuclear reactor. And what you're doing with neutron activation analysis is you're bombarding your sample with neutrons. And if you remember, this is like from Gen Chem, if your atom has a different number of neutrons, then it's a different isotope. So a very simple example is hydrogen, deuterium, and tritium. Hydrogen, the most common type of hydrogen is just the proton in the nucleus. Deuterium has twice the weight because it has a proton and a neutron in the nucleus, and they weigh about the same. Okay, and tritium has 2 neutrons and a proton in the nucleus. Tritium tri 3. So it's 3 times the molecular weight of a regular hydrogen, which is just the proton. So those are just different isotopes. The thing is, when you slam neutrons, first of all, it's not easy to slam neutrons into a nucleus. Most of what you learn in chemical reactions involves, oh, the electrons got, transferred, and you have an ionic material, or the electrons were shared, and you have a covalent bond, and maybe depending on the different atoms, some of the electrons like to be closer to this atom as opposed to that atom, so you have a little bit of charge differential between in that bonding. So what neutron activation does, it requires a lot of energy to slam neutrons into a nucleus to create an isotope which is radioactive. And what happens was when you create a radioactive isotope, it'll give off gamma rays, so really short wavelength energy. And the wavelength of that energy will be consistent with the original atomic structure. So you can determine what elements were originally there by which gamma rays are being emitted by the isotopes that are produced with neutron activation analysis. So neutron activation analysis used to be one of the methods that they used to examine hair. It's extremely sensitive, so you don't need an awful lot of a particular type of atomic material because you can detect the gamma rays that are coming off from that particular, those atoms inside of your sample. But yeah, so something like this, you can't distinguish between, came from outside, came from inside. Neutron activation analysis for a short time in forensic history was used to try and individualize human hairs by saying, okay, the crime scene hair had this elemental profile, and the known hair also had a similar elemental profile, trying to do that. But statistically, it did not work well because typically crime scene hairs, if you only have one of them, it does not give you enough statistical support. Usually, good statistics, you have several samples to look at, and if you only have one, then you have a lot more error associated with that analysis. Okay. We already learned about those 3 growth phases, anagen, catagen, and telogen. So anagen, actively growing, telogen, not growing at all, and catagen, the transitional phase from anagen to telogen. And how much does it grow? About 3 millimeters a week, or it depends on the person. So some of it's genetic, some of it's how good your diet is, if you're healthy or if you've got a cold or if you're dealing with some kind of illness. Those things can all affect how much hair growth will happen. So anagen phase, squishy end over here where it's not keratinized yet. Phase of active growth is depending on the person, genetics, diet, and environment between 2 and 7 years. You can see over here a follicular tag. Sometimes you'll see that if a hair gets plucked out, and the sebaceous oils, which are somewhat waxy, are on the inside surrounding the hair, and sometimes it comes out with the hair, and that's your follicular tag, and sometimes people don't have that because it stays inside of the skin. Okay. So sometimes you see a follicular tag, and sometimes you don't. Catagen, the follicle decides it needs a rest, and it'll begin, the hair will begin contracting. The follicle will no longer have melanocytes pumping in melanosomes, so when the hair begins to go into resting phase, it'll start to look pale. So you might have a black hair, but at the root, it's not dark, okay? And definitely by the time you get to telogen phase, you've got like a white root, even if the hair itself is dark. Okay. So this will usually last a couple of weeks. You begin to get the bulb shape, and then by the time you're done, you probably could, I could probably like find like a few of my hairs coming out real easy right now. And if I look at the end of it, it'll have a round bulb that we're just holding in place. It'll be intelligent phase. The hair will eventually fall out, maybe for several months before it kicks back into anagen phase. And most of your hairs are in anagen phase. The follicles that begin to go into resting phase, randomly throughout your hair, depending on how long that hair has been growing. That's why it's not like all your hair falls out at once and then all of it starts growing. Yeah, that does not happen. Some body area hair cycles. So antigen, catagen, antigen intelligent actually. And you can see over here, on your scalp, 10 to 15% of the follicles are intelligent stage. Most of them, therefore, are in antigen and maybe even smaller quantity were in catagen phase. Okay, and some of it we don't have information, but you can see there's like scalp, eyelash, eyebrow, mustache, beard, chest, axillary. Axillary? Actually, it's a good question. I think someone look up axillary. You'll have Google, right? I don't know.
Armpit.
Armpit. Okay.
What a fancy name for
Armpit
that.
hair. Okay. Oh, look, they don't even have information about antigen intelligent over here, even though they do say 31 to 79%. It's a big range of armpit hairs that are in intelligent phase. But hairs all over the place, and they don't even have antigen intelligent information for the pubic hair. But look, a lot of it's in intelligent phase, so you have a lot more turnover in the pubic region. Okay. Intelligent hair is commonly encountered because they fall out without you even keeping track of them. But we cannot specifically determine the age since detached. You can only use contextual information to say, oh, it's really dirty and dusty. Maybe it's been there long enough for little bugs to eat it, to use it as a nutritional resource. You will see some hairs that are old that it just looks like a bug bite in it or chomped away. So yeah, bugs will eat. It's protein, right? If you're looking at a question hair, definitely look at the root. Is it antigen, catagen, intelligent? And if you have a hair from a possible source or representative sampling, if you have an antigen hair, you're going to want to look for an antigen hair from the representative sampling to do a direct comparison of roots. Obviously, intelligent hair from the same person will look different from the antigen root, right? Because it's just different characteristics. So you want to compare. And when you do that comparison, it's best, it's not always possible, but it's best if it's from the same time frame. So if you don't arrest the suspect until a year or two later, then okay, you get the hairs that you can, but it's clearly a different time frame from if that person really is a perpetrator 2 years ago versus now. There might be some different characteristics over time. Okay.
So how would you compare those two samples? Like if you had...
You still could, but you would have to be more careful in the conclusions you draw. There might be some differences, for instance. Let's say, I don't know, let's say the person went vegan in that time frame, and so their diet changed. No, I'm not kidding. You know, it's like when your environment, when your, I guess the nature's not going to change because your DNA is your DNA, but your nurture might change, your environment, your diet. You used to exercise a lot, but now you're a couch potato. Like those kinds of things will affect biochemicals throughout your body. So you might see some differences that are a result of environmental differences or just changes in behavior. And if you also have a time frame difference, then you can't say as much about the sample. I use the same example like a footwear imprint. You got a nice detailed footwear imprint at the crime scene. The guy was wearing Nike sneakers and walked around, stepped in a pool of blood and left like a big trail of footwear imprints. And there were a lot of defining characteristics like wear and tear marks. And you're like, yeah, if we can find the sneaker, there's a lot of information to do a comparison. But then the guy doesn't get arrested until a year later, still has the same shoes maybe if you're talking about the actual perpetrator, but he's been running around in those. And now those wear and tear marks have shifted. Some of the things you saw a year ago are no longer there, and some new marks are there, which means that class characteristics are the same, shoe sizes, that kind of thing, but you're not able to do a direct comparison and say I've individualized, but there's a reason for that. So you're not going to necessarily say I've individualized, but you can still say, well, it could have come from the same shoe, but the time frame and the continual usage has caused differences over time. So that's like the shoe analogy to how your hair might be different, your root might be different. Over time, it changes in your nutrition and your behavior patterns. clumps of hair that are anagen, like I said, maybe someone's fighting for their life and grabs onto someone's hair and rips to defend themselves. But like I said, if you only see one or two of them, then that doesn't really, it's hard to, most people don't pick one hair to fight for their life. So don't do that. When you're collecting hair evidence, this is straight from FOS 313, like the first few things we talked about. What can you see? What can you say about the context? You'll get some hairs from tape lifting, some from vacuuming. Sexual assault evidence kits, in addition to doing things like a vaginal swab, perhaps a rectal swab, perhaps an oral swab, depending on the nature of the assault. They will also collect hair samples from the victim and try to distinguish hairs that are foreign, so more likely to come from a perpetrator versus hairs that are her hairs or his hairs, depending on the victim. So that representative sampling is key to being able to understand these things. You've already done these things, like the species of origin. You may have had a chance to look at different somatic origin hairs because we had those in the 313 lab. So all of these are things that we're trying to figure out. You already know you mounted fibers last semester, and this semester you'll definitely be mounting your unknown hair. So I would recommend you take some practice and mount your hair. It'll be kind of cool to have your own hair, and then you can show your mom or your friends, mounted my hair. And so under those conditions, I know we used to have big slides with big cover glasses, cover slips, because if you have something like this, it's a pain in the ass, I tell you. Try and mount this on a regular small slide, even with the longer 50 millimeter cover glasses, cover slips. With something like this, I would typically do concentric circles. I don't like doing figure 8 because they cross over each other and you have to smush it down. But if you do concentric circles, and it's not easy to do this. And I'll tell you my tips for doing that later on if you're interested. Okay. All these different things that we've already learned about, and I know we're closing in on time. SWGMAT, Scientific Working Group on Materials. One of many things that they look at is hair. I don't know if this link is still active, but if you look up SWGMAT hair, you will find the standard methods that hair analysts use for analyzing human hair. I would suggest that you take a look at that and you can incorporate that. One of the nice things that we started doing a couple of years ago is we permit you to use basically data sheets for each hair that you look at. So you can get the data sheet, it's in your lab manual, and fill out for each hair that you examine so you have information about that particular hair. We've done this already. This is an example from Petracco. It's Nick Petracco, the same Nick who will be the occasional Monday lab instructor along with Dr. DeForest. You saw this in 313. We talked about this, and you can do this as well in the Crim Lab now. We do have clear nail polish for you to do the scale casting. And if you have maybe a pet dog or a pet gerbil or whatever, please do not rip hairs out of your pet. But if hairs have fallen out naturally, you can also do mountings of their hair and do scale casts to see that your dog hair, the cast looks different from your hair, that kind of thing. Please do not torture your animal. And all this stuff is, like I said, all from FOS 313. Just some of the things like sometimes you'll see bucklings, changes in this kind of thing is similar to when you had to estimate the thickness of the fiber last semester. Really dark hair. Weird, right? Undulating shaft. Convoluting, like it's twisted. Twister kink. Abrupt diameter change with a lot of air in the medulation. And I mentioned the quarter wave plate, and that's how it looks. See, the cuticle is really easy to see when you use a quarter wave plate in a subtracted orientation. Really good contrast. So if you're having difficulty seeing it under ordinary, not in cross polars, then you can add your quarter wave plate. And then other things, We talked about cross sections before. And I think this stuff I've alluded to. Like I said, if you see insect bites, if you see a dusty hair, there's an indication that it's been there a while for that to happen. Okay. Mechanical damage, well, I mean, I have some split ends. Mechanical damage will depend on the degree of damage you see. For instance, it looks like the hair has been stepped on, and the degree of damage is more than you would ever expect if the hair was on someone's head, unless someone was being abused, and, I don't know, someone was stepping on someone's hair that was already on their head to keep them in place, for instance. And I'm not, so there's dirt on the hair, broken hair, mechanical damage, even more damage. And I'm not even going to go into post-mortem root banding, but if you're interested, I will talk about it outside of class time. But what does it look like? That is an example of post-mortem root banding, even more advanced. And sometimes this decomposition in a hair that's in a decedent or a dead person advances to the point where if it gets pulled out, part of the hair stays in the scalp and it's on brushy. Okay. But post-mortem is after someone's died, and root banding is this dark band that forms. And it's another interesting thing that we're still trying to figure out. So you guys can run off and have fun and laugh later today. Some of you came in later. It's also on Blackboard, but the exam schedule is up here if you want to grab a copy of that. And I did see that I got, I can add information today. I'll email you, and you can go in and thank you. OCME biology, right?
You want to? No.